Molecular imprinting is a technique developed by polymer technology to create template-shaped cavities in polymer matrices with predetermined selectivity and high affinity. This technique is based on the system used by enzymes for substrate recognition, or cellular receptors for signal recognition, which is called the lock and key model. The active binding site of an enzyme or receptor proteins has a shape specific to a substrate. Substrates with a complementary shape to the binding site selectively bind to the enzyme. Alternative shapes that do not fit the binding site are not recognized. Molecularly imprinted materials are prepared using a template molecule and functional monomers that assemble around the template and subsequently get cross-linked to each other. The monomers, which are self-assembled around the template molecule by interaction between functional groups on both the template and monomers, are polymerized to form an imprinted matrix, commonly known in the scientific community as a molecular imprinted polymer or MIP. The template is subsequently removed in part or entirely, leaving behind a cavity complementary in size and shape to the template. The obtained cavity can work as a selective binding site for the templated molecule. Various type of approaches such as covalent, non-covalent, ionic/metallic etc. were developed in molecular imprinting technology. One application of molecular imprinting technology is in affinity-based separations for biomedical, environmental, and food analysis. The feasibility of MIPS in solid-phase extraction, solid-phase microextraction, and stirred-absorption extraction has been studied. Chromatography techniques such as HPLC and TLC can make use of MIPS as packing materials and stationary phases for the separation of template analytes. Another application is the use of molecularly imprinted materials as chemical and biological sensors. Molecular imprinting has steadily been emerging in fields like preparation of artificial antibodies, drug delivery and biotechnology. In the biopharmaceutical market, separation of amino acids, chiral compounds, hemoglobin, and hormones can be achieved with MIP adsorbents. MIT uses the concept of molecular imprinting and molecular imprints to explain homeopathic potentization in this scientific perspective. During potentization, three-dimensional conformation of drug molecules are imprinted as nanocavities into the hydrogen-bonded supramolecular networks of ethyl alcohol water matrix. These molecular imprints can act as artificial binding sites for the drug molecules used for imprinting, as well as to pathogenic molecules having similar configurations.